DNA structure and discoveries. We're going to talk about a series of people who figured out things about DNA. These are the cool dudes of DNA and ladies. So we're going to start off with Frederick Griffith. And he was trying to find a vaccine for pneumonia, and the bacteria that causes that is Streptococcus pneumoniae. Notice it is italicized because it's typed. So he's trying to find this vaccine. And he finds that there were two strains. The rough strain, which is non-pathogenic, and the smooth, which is pathogenic. And it looks smooth because it has a capsule, which usually means it's pathogenic. And so he injected each into mice. R cells, mouse is okay. S cells, death. And so then he heat killed some S cells and injected them into the mouse. Mouse is okay. And then he heat killed S cells and mixed them with living R cells, and the mouse dies. Oh, I'm sorry, I forgot the sign for death. So you're saying, what? How is that possible? Well, cells can actually ignore that. There's this cool thing called transformation. And it's this idea that living cells can actually pick up DNA from their environment. So these non-pathogenic cells can pick up pathogenic genes and pretty much change their genotype and phenotype and become pathogenic. Oswald Avery spent 14 years trying to figure out what this transforming agent was, because they didn't know it was DNA. And so they knew it was either DNA, RNA, or protein. And so what he did is he broke open heat-killed bacteria and extracted cellular components and activated one of each of those three, the DNA, RNA, or protein, each time, and then tested to see if the heat-killed bacteria could still transform non-pathogenic bacteria. And only when DNA was active did transformation occur. So if it was inactivated, it didn't happen. So DNA must be the transforming agent. All right, hang on. All right, now Alfred Hershey and Martha Chase were looking at viruses. And there are these T2 viruses which infect E. coli. So you can see these the little viruses on the bacterial cell here. And they already knew that T2 was made of DNA and of protein, and that T2 turns E. coli into a T2, this is tongue twisting, T2 production factory. And so the way they went about this is they took the bacterial cell here, and they took those viruses and put radioactive sulfur into the capsule, and then they put radioactive phosphorus into the DNA in another set. Same bacteria. Let them grow in culture, and then pretty much blended it up to separate all the cellular components, and they found most of the sulfur outside the cells, but they found the phosphorus inside the cells. So only DNA actually enters E. coli during an infection, and therefore DNA is the transforming agent again. Edwin Chargaff, already knew that DNA was made up of nucleotides. And a nucleotide is a pentose sugar or a 5-carbon sugar. So they look like little home plates. A phosphate group and a nitrogenous base, either adenine, thymine, cytosine, or guanine. Find those spellings in your book, or I'll give them to you in class. And he compared, the chemical composition of different organisms' DNA, and he found that the composition of the bases varies between each species, and within a species, the percentage of A equals the percentage of T, and the percentage of C equals the percentage of G. And like in humans, you have 30% A, 30% T, approximately 20 C, 20 G. Rosalind Franklin, oh look, another girl. She used X-ray crystallography to look at DNA, and this is her lovely picture of DNA. And it confirmed that it was a helix. And then a lot of people were working off of her unpublished paper, and they knew that the sugar phosphate was the backbone. And then you have these guys, Watson and Crick. And Watson and Crick came up with the double helix, the model of DNA that we know today. They know that it's 2 strands, sugar phosphate backbone, nitrogen bases are hydrophobic, therefore they need to go interior. It's anti-parallel, which means when you look at this little home plate over here, I'm over here, this little home plate versus this little home plate, they're upside down, right? That's anti-parallel. You get a full turn every 10